Hi, welcome. We're talking about BT430. We're talking about metagenomics and extremophiles. You have to excuse the noise in the background. Not much I can do about it. Okay, so in this course, we're looking at a lot of micro DNA and trying to figure out how to look at these organisms that we call extremophiles. We're also looking at the importance of metagenomics. Why is it that we needed this whole new science in order to understand them? And we'll look at that and look at case studies about them. But before we head into metagenomics, we're going to first and foremost just look through the course outline because what we have to first of all look at is the diversity of extremophiles and how we handle them, how we search for them, and their diversity. Then we can start looking at the DNA and the topics that are really related to metagenomics, which is working in the DNA of many organisms at the same time. I have to update these dates so that they're actually valid. But we're supposed to talk about extremophiles, then our applications, and I gave you some handouts to that effect before we left. Then we're discussing the diversity. How do we know what kind of extremophiles are really out there and how do we study them. Then we talk about the application of phylogeny to understand how the species are in an environment. So when we talk about species richness, we're talking about the abundance of different species depending on that extreme environment. Then we also use specific molecular markers to understand these different organisms because it's very difficult to actually culture them most of the time. So we depend heavily on DNA and molecular markers. And then we'll compare the cultivation techniques for actually working with some of the extremophiles, but you'll find that mostly we have to use molecular techniques to assess the diversity. Then we look at the different databases that we use for extremophiles and metagenomics. So some of these will you draw on the knowledge that you got from BT325, where you learned about using biological databases. So there's NCBI, and then there's a whole bunch of specialized databases that we use to understand extremophiles in particular and how to handle metagenomic DNA. So we'll also have a quick look at the actual software we use. Never mind the noise in the background. There's nothing I can do about that. Um, then we have, um, when we look at the classifications, I already gave you the different groups of extremophiles that we think about, and you should be familiar with those, but we take a deep dive and look at the different classes that there are, and also compare it to the African situation. Where do you find them? Where do they exist? What does it mean for biotech? Then we'll look at the physiology and biochemistry. So how do these organisms actually manage to do what they do? And we look at specific adaptations. So that's the proteins, the membranes, and different kind of strategies they have to survive in the conditions where they survive. And this will draw on your biochem and your use of databases again, NCBI, all those places to get sequence data and things like that, and also the 3D structural data of these different proteins. Then we'll look at other strategies like compatible solutes, lipid bodies, DNA replication, DNA repair. So these are some of the challenges that the organisms have, and these are the adaptations that they found to overcome those challenges. And then we'll look at the techniques that there are to increase the cultivability of extremophiles. So it's difficult to culture them, but you could do one or two things to try to increase your chances. Then we also look at how we sample for these organisms, how do we get them in the environment. And here we look at how we extract the DNA. So before we talked about DNA extraction, just general, but when you're trying to extract it from these organisms, usually the samples are they're contaminated with things that go against PCR and things like that, that actually impede PCR. and other downstream processes. So you have to have special techniques to extract DNA from these organisms so that you can use it for other purposes. So that's what this section is all about, how to extract DNA, RNA, and proteins, even lipids from these same extremophiles, and also the enzymology. Then we look at biogeochemical cycles. So this is when we look at how these organisms participate in the cycles of life. So you've heard of the water cycle, you've heard of the carbon cycle, and all these other things. Biogeochemistry looks at the cycling of these nutrients, the cycling of phosphorus, of nitrogen, of sulfur, and what role these organisms have in that. So we'll look at that and see what happens if they're not there. Then we look at the actual genes, which are the markers of these cycles. We see that we have to use molecular biology a lot in order to work with extremophiles. Then we'll think about doing like a poster presentation to show what you've understood. And then finally, we start going into the metagenomics. So how do you construct a metagenomic library? How do you discover new genes using metagenomics? How do you generate sequence from your metagenomic experiments? How do you actually get high molecular weight DNA, which is what you use to work with metagenomic DNA? How do you construct these libraries? Part two. So this is part one, where we just sort of understand what it is. Over there, here we actually talk deeply about it. And then how do we screen these libraries that we've created in order to find something of use? And that's basically it. So those are the topics that we'll try to work through. We'll see how far we'll get. So to get you started, let's look at the notes on the biodiversity, right? So we started talking about the diversity of these extremophiles. What are they? And where can we find them? What's the point of all this? So extremophiles are organisms that thrive in extreme environments. And they're only called extremophiles if you're looking at the situation being detrimental or lethal to most forms of life. So these organisms live in places where normal organisms can't. And they've developed very special adaptations to do so. And so we categorize them based on pH, salinity, pressure, radiation, and there's also other things to do like water availability and such. So quickly, this is just a snapshot of the extremophiles that we generally deal with. We've got thermophiles, so these are the ones that like high temperatures. We have hyperthermophiles, these are the ones which are even higher, so really high temperatures. We've got psychrophiles that love the cold, halophiles that love high salt conditions, acidophiles that love acidic conditions, and alkaliphiles which like alkaline conditions. Barophiles that love high pressure environments, and xerophiles that love really dry conditions where you have very low water availability. And then we've got metallophiles that love high concentrations of heavy metals or metals in general. They don't have to be heavy metals. And then radiophiles that love radiation. So these are the different ones that we'll be working with and thinking about and talking about and examining. Now you can imagine in these different conditions, all of these pressures make it difficult for biological life to proceed. So from your biochem, you must understand that there's limits in which these biochemical reactions can take place, right? So if we're talking about temperature, there's limits before proteins denature. If you're talking about the other side of temperature, there's limits at which the liquid in a cell, because the liquid, the water is for the solvent to try to like make all the chemical reactions happen within that solvent. Now, if it's frozen, what are you going to do? So you've got implications of temperature, then salt conditions. Also, that's to do with osmosis and so on. So if you have high salt conditions, that affect osmotic pressure in the cell, especially in plant cells where you have, that plants wilt if they don't have enough water. So even the ability to be turgid, to be stiff, is difficult because of just having too much acid or too much salt or so on. Then you got acid and alkali conditions. Again, this is a lot to do with transport of things across the membranes of the cells. And there's other issues that come up when you start changing the amount of protons available or the amount of hydrophilic molecules available. This is a problem, but they have to learn how to survive. Then you got pressure. What do you do when you're not at atmospheric pressure like where we are at sea level? If you go under, like go down deep into the sea, then you've got a very big problem of pressure. Things just being squashed like this. How do you live when you're busy being pressed down on every side with very high pressure? And xerophiles, what do you do when you don't have water? We know that life needs water, liquid water, to survive. What do you do when it's not there or it's very low? And metallophiles, these also interfere, the metals interfere a lot with biological processes. You know, in your hemoglobin, you've got iron there. That's doing its job. What happens when you have too much? You've got loads of other metals, and now it becomes a problem. It's not even helpful anymore. And then you've got radiation. This is also a big problem because it means that you have mutations happening in your DNA, amongst other things. So it becomes a problem if you're exposed to these things. But somehow these organisms have found ways to counter all of these issues. And that's why they're fascinating. And these are the environments where you're going to find them. So you're going to find them in places where when you walk past or go there, you think there's nothing. So places like hot springs, you think there isn't, especially if it's very hot, you think there's nothing growing there, or hydrothermal vents, which are in the bottom of the sea, or in the deep oceans where the pressure is so much and it's so dark, you wonder how anything will survive. The deserts, there's hardly any water, high altitude environments, and brine, soda lakes, nuclear reactors, ice sheets, polar regions, toxic waste sites. These are all habitats for extremophiles. They love it. You and I can't manage that, but they absolutely love it and they thrive there. And so our job is to understand how, understand the mechanisms, and see how we can actually study them. How do you study them in the lab in order to uncover all of these mechanisms? So extremophiles represent a significant portion of microbial biodiversity, and they encompass the whole domains of life. That's bacteria, archaea, and eukaryotes. So this is now coming to phylogenetics. Um, You remember last year when you had to choose which vegetables you wanted to pick for your biotech? We are doing it according to different plant families, isn't it? This is a type of phylogeny where you're grouping things according to how they're related. So these would all form branches on a phylogenetic tree. Now, when we're talking about the tree of life, we have a tree that looks more like this, and we talk about the different domains. And the phylogenetic tree of life, it's got certain features, and we'll go into more detail about this, what it means, and also how you can prepare it yourself when you have sequences. So here you have the root of the tree. So it can be rooted or unrooted. There's some trees which are like this, where they're not rooted, and then other trees are like this, where they're rooted. And we find out more about what all this means when we start constructing our own tree. But when we talk about the three domains of life, we're talking about the archaea, then we're talking about the eukaryota and the bacteria. And when you look at this tree of life, you'll find that the archaea were there first, and the bacteria, or the not bacteria, sorry, the uh, the prokaryotes, and then the eukaryotes. So that's how it goes. And this tree of life allows us to sort of have a snapshot at how these things develop. So you can ask yourself, well, how do we come up with this tree? There's this notion of the molecular clock. So this is the molecular clock hypothesis that talks about how mutations in DNA can actually be related to time spans. So the more mutations you find, the longer time has actually passed between when those mutations came. And you can relate them all in a tree like this to be able to see when something emerged first or when something emerged later on. And when we talk about this molecular clock hypothesis, then you will start to understand that when you look at a phylogenetic tree, it has a root, it has a source, it has a beginning, it has a developmental pathway. And then the latest ones are at the tops of the tree, like that. So the earliest ones are down at the beginning. But you'll get how to read that once you understand what a molecular clock is and how we use that in order to infer time spans when we look at phylogeny. So phylogeny helps us to do a couple of things. It helps us to understand how things develop. It helps us to sort of put timelines to it and also to see how things are related. So when we're talking about phylogeny, this is what we're talking about. And the whole point of it is to produce these kind of trees that show how different organisms are related. And we generally use DNA or RNA or protein to generate these trees and bioinformatics. Okay, so back to the diversity. So that's about bacteria, archaea, and eukaryotes. The discovery of extremophiles has expanded our understanding of biodiversity and the limits of life. Many extremophiles exhibit remarkable adaptability and resilience. This is why they're so important to biotech. They showcase the evolutionary processes that allow life to thrive under extreme conditions. So when we look at extremophiles, we get a window into how the Earth was and how life can be beyond the Earth in other places where these conditions might be the norm. So if you look at the history of the planet, it was a hot place first before it cooled down to this temperature. Even the environment, the atmosphere used to be a reducing environment. Now it's an oxidizing environment. And so what you find is that there are certain organisms in these groups that developed when life used to be very different here. And as things changed, then you have the evolution, as it were, of organisms that are more adapted to the way that the planet became. So when you look at the diversity of these organisms, you actually see the story of life on Earth. So that is what this course is all about. And why is it biotech? Because all these adaptations, they mean source of genes for us. So if we want an organism that can do things that normal organisms can't do, that's why we go to extremophiles. If we want something that can tolerate high temperatures, we look at thermophiles. We find out how they do that. Then we get the genes from there and we put them into an organism that we want to be more tolerant to high temperatures. Or when we're studying thermophiles or hyperthermophiles, how is something able to have DNA reproduction in boiling water? It's just an extreme thought because you know that there's coagulation, there's denaturation of these proteins, but how does this organism do it? So when you start looking at the proteins, how they've developed, you see certain patterns, certain motifs that it uses only certain amino acid or it uses only certain structural motifs in order to make its protein so that they don't denature quite so easily. So that is why we look at it. That's why we use extremophiles. They're such a great resource for biotechnological tools, really. So whether it's genes, whether it's proteins you're looking at, or just new ways of doing something. So new ways of actually building in synthetic biology, where you're building proteins from scratch and you're actually synthesizing these proteins. Instead of you doing trial and error, you can look at an extremophile and see, well, this protein or the hemoglobin, is able to withstand this temperature because it has this certain, like a loop or alpha helix, or it just uses beta pleated sheets, all that kind of thing. Or it just uses certain codons in its DNA. All those things become obvious when you study these extremophiles. And that's what allows you to be able to now take that learning and apply it in a biotech kind of solution. So that is why these organisms are so important. And it's our job to understand how we classify them. How do we talk about these domains of life? How do we investigate them, the tools that we use? And then we need to also understand this metagenomics. This is so important. So here we talk about how do we study the DNA of many organisms at the same time. Usually, when you did your analysis of DNA, it was just gene by gene from one organism at a time. But when you're studying these organisms, because they usually exist, in environmental conditions, right? Maybe it's in soil, or maybe it's a metagenome inside the gut of an organism, or something like that. They usually occur in groups. So you have to study them together in groups. So instead of studying just one uh, DNA sequence at a time, you have to study them all together. And that's what metagenomics allows us to do. So that is what we'll be doing. And I would love for you to just revise on these different organisms and start thinking about conditions in Zambia where you could find them. All right, that's it for now. Thanks.